Namaste. Welcome to this 5 days national workshop on CSR, UGC NET and LS preparation in life sciences organized by Botany for You. At the onset, I thank Dr. Pitambar for organizing such a huge opportunity for all the aspirants of the NET throughout the country. Now, the topic which is given to me is cell signaling, and I will take this cell signaling from competitive exam perspective so that you will be more aware of what kind of questions are being asked, and if you are looking into the cell signaling, what are the things that you should take care. So let's start about the cell signaling. Yeah, so the cell signaling is a orderly sequence of events by which a cell communicates to another cell and to its environment. So this entire process by which cell interacts with one another and their environment so as they can regulate various metabolic processes, cellular processes like metabolism, migration, differentiation, and even the apoptosis or cell death. The signals for all these regulatory pathways are being governed by various cell signals which are conveyed from the cell to another target cell. As you can see on the slide, when there are two nearby or neighboring cells, they can communicate to each other by means of gap junctions through which certain ions or certain signaling molecules can be given to a cell or can be received from the neighboring cell. But for a far distant cell, because with the advent of the complexity, various cells which are distributed here and there in the body, they need to communicate with each other. Now, this communication is governed by cell signaling pathways. Now, a particular cell which needs to communicate to another cell will secrete or produce a signaling molecule, which is also called as ligand. Now, this ligand, that may be a hormone, that may be a neurotransmitter or a growth factor. Chemically, they can be ions, they can be peptides, and they can be even gaseous molecules which will be released by one particular cell, and the same ligand or the signaling molecule will be received by the target cell, because the target cell at that time will be having a receptor for that particular ligand molecule. We will see about this entire thing. Now, as far as signaling molecules are concerned, they are synthesized and released by the cell which needs to communicate to another cell. And whenever that particular signaling molecule will be received by the target cell, that will bring about the effect. That will do whatever the message has been given. So this primary signaling molecule is also known as primary messenger. Now, the essence of cell signaling can be seen in this particular slide. As you can see, a signaling molecule or ligand will bind to the receptor. Now, these receptors in most of the cases are present on the cell surface, but receptors may be cytoplasmic or cytosolic. That is, receptor may be present within the cytoplasm of a cell. In this particular case, we just need to look on the entire cascade. That is, this signaling molecule, it is interacting with the receptor. Now, there occurs a very high affinity between the ligand and the receptor molecule. And once this particular signaling molecule is received by the receptor, what will happen? It will bring about a conformational change in the receptor. And then receptor will convey the message via a variety of signaling molecules. These might be certain kinases, these might be certain enzymes like adenylyl cyclase. We will discuss about it in the later part. So ultimately, this entire message is relayed by a variety of players or signaling molecules. And finally, how the effect will be brought about? The effect will be brought about by activation or inactivation of certain enzymes, by phosphorylation, dephosphorylation, or fragmentation, or their synthesis, or by inhibiting the synthesis of a particular metabolite or hormone, by gene modulation. That is, one or the other gene may be activated or may be inactivated as a result of this entire cascade. Now, cell signaling is very important because it provides a means by which the organism interacts with their environment. Is there any toxic molecule around the vicinity of the cell? Is there is a scarcity of the nutrient around the cell? All these signals, including various abiotic signals, are being conveyed by environment to the cell, and these messages, these signals are received by the cell so that it can act, so that it can respond accordingly. Now cell interacts with each other to bring the necessary effect or the response in a particular situation. So in a particular situation, what particular kind of enzymes needs to be activated, which particular proteins needs to be produced, all these things, all these signals, all these tasks are being conveyed by cell signaling cascades. It's a mean of attaining the coordination and regulation. In addition to the nervous system, this is one of the finest mean of attaining the coordination among the various cells of the body and to regulate various cytological processes, various physiological processes. Cell signaling is very much mandatory. By means of cell signaling, various metabolic, physiological, or molecular processes are controlled, whether it is replication, whether it is transcription, translation, various pathways of metabolite anabolism or catabolism. All these things are governed by cell signaling pathways. Now, the interpretation of the environmental cues, that can also be described at the cellular and subcellular levels by means of cell signaling. So, in a nutshell, it is very important and crucial, and it plays a central role in the various aspects of the cell. This particular slide that we have already seen earlier, again reinforcing the same fact that first thing, reception of the signal, there comes transduction, that is passing or relaying the molecule, and then finally the effect or the response by activation of certain cellular responses, and that activation is governed by gene modulation or enzyme modulation, that is switching on or switching off of a particular gene or activation or deactivation of a particular enzyme. That is how the desired effect or response will be brought about. Now, if we so far we have discussed that cell signaling, the cell that needs to give a message to another cell will produce a signaling molecule. Now, these signaling molecules can be intercellular or they can be intracellular. So, in multicellular organisms, variety of signaling molecules are there in terms of chemical nature. They can be amino acids, they can be lipid derivatives, they can be peptides or proteins. All such molecules are being used in the cell signaling pathways by our body. There are certain molecules which are intracellular, that is, they may enter into the cell. Obviously, one thing plays a very important role, the nature of that particular chemical. If it is lipophilic, it may enter into the cell, and that signaling molecule or that ligand molecule will be, you can say that, intracellular one. So, this is very important fact that steroids, retinoids, and thyroxine hormones, they all are lipophilic or hydrophobic. So they may enter into the lipid membranes of the cell without any problem because they are lipophilic, and they can enter into the cell. So obviously, such ligand molecules will be having intracellular receptors present in the cytoplasm, and in some cases even in the nucleus of the target cell. And that is why questions are being asked on this particular aspect. We will be discussing such examples in the later part. Now, if we want to have an overview of the entire signaling pathway, so it starts with a particular extracellular signal, which is called as ligand or signaling molecule. So far we have discussed it can be intercellular, it can be intracellular. Chemically, it can be very diverse. Sometimes they do ask on the chemical nature of a particular ligand molecule, particularly those which are gaseous like NO, those which are ions like calcium ions, they all are signaling molecules. And since they are unusual, questions are asked on that. There comes receptor, because ultimately a ligand released by a signaling cell will be received by the target cell by means of a receptor. So that particular receptor may be cell surface receptor in the case of lipophobic or hydrophilic ligands like various peptides, proteins, and it can be intracellular in the case of various steroid or other lipophilic ligands. There comes another category of signaling molecule that is known as effector proteins. Now the effector proteins, they are certain enzymes which are membrane bound, and they will bring about the synthesis of the secondary messenger. We will be discussing what the secondary messengers are, but there are certain effector proteins which are activated and they will generate secondary messengers like cyclic AMP from ATP, cyclic GMP from GTP, and so on. And the secondary messenger plays a very important role. Now, what is the concept of secondary messenger? The primary messenger is the ligand molecule. As you can see, this red dot, this is signaling molecule, and this particular messenger is known as primary messenger, but it is mostly outside the cell. Within the cell, whatever needs to be done, that is being governed by the secondary messenger. So coordinator within the cell, which is doing variety of things so as to bring about the desired effect, will be governed by secondary messenger. These secondary messengers are generated from effectors. The examples of secondary messengers are cyclic AMP, cyclic GMP, diacyl glycerol, inositol triphosphate, calcium ions. So sometimes they do ask which of the following is a secondary messenger, and sometimes they do ask the full form of IP3, for example, inositol triphosphate. So all these things are normally being asked, and then the secondary messenger will bring about the effect by means of various protein kinases in the cell. Here I should mention that phosphorylation and dephosphorylation events matters a lot in the cell, because various enzymes, various transcription factors, they are switched on and they are switched off by means of phosphorylation and dephosphorylation events. And whenever phosphorylation of the substrate is required, it will be done by one or the another kinases. Not only one or the another kinases, sometimes there is a cascade of kinases, as we will see later on. So protein kinase A, protein kinase C, protein kinase G, and almost 700 different kinds of protein kinases are there in the cell, which reiterates the importance of the kinases in our cell and signaling cascades. Now, when a particular protein or particular transcription factor is activated by phosphorylation or inactivated by phosphorylation, in that case, one particular process, one particular response can be switched on or switched off, can be speed up or it can be lowered. So that is all being done by protein kinases. Now, if we see the signaling overview, as we have seen in the previous slide, the signaling molecule will act as a ligand, which binds to a structurally complementary site on the extracellular or membrane spanning domain of the receptor. So ligand receptor interaction is a bimolecular association reaction, which have a very high affinity and very high specificity for each other. So it will never happen that a particular ligand which is being produced for a particular effect will not be received by any another receptor, because there occurs very high specificity and very high affinity of ligand for the receptor and vice versa. So whenever a ligand will bind to a particular receptor, then only the desired effect on the receptor will be bring about. For example, it might be a conformational change. Because of that, it may become accessible to some other signal transducing molecule, or it may become amenable to phosphorylation by some other unit or some other signaling molecule. We will see later on. So broadly, if we look into the types of signaling on the basis of the distance over which the signals are being governed, so sometimes they do ask questions on these things, although it is very basic thing. So when the signaling cascade is on the same cell which is releasing a particular signaling molecule, and the same cell is receiving the ligand because it is bearing the receptor as well, so then it is called as autocrine. You can analogy can be taken as self-talk. When we talk to ourselves, we are speaking and we are receiving. So that kind of thing, when a cell does, it is called as autocrine signaling. When a cell is communicating to very nearby cell, then it is called as paracrine. When a cell is doing the communication, or it is conveying the signal to the adjacent cell, then it is called as juxtacrine. Here comes affix. Paracrine and juxtacrine, in both the cases, the signaling molecule is traveling a very less distance, and the cell which is producing the signal and cell which is receiving the signal, both are very much close to each other. But adjacent is more closer. So juxtacrine means very nearby, just almost in touch. And here the receptor present on one cell will interact with the receptor present on the another cell. For that, these two cells have to be very, very closer, even more closer than that of paracrine. There comes endocrine, that we have studied in the case of hormones, that a particular signaling molecule will travel a long distance so as to bring about the desired effect. There comes synaptic, which occurs at neuromuscular junctions or the synapses, brought about by a neurotransmitter acetylcholine. And because of this particular fact, there are questions on acetylcholine. Now, this is the diagrammatic representation of the signaling molecule which is occurring at synaptic signaling. And you can see here the same cell which is producing it and it is receiving it, then it is called as autocrine. When it is being given to the nearby cells, it is called as paracrine. When the signaling molecule travels a large distance via the blood vessels to reach the target cell, then it is called as endocrine signaling. So these are the various types of signals. The most important fact here to remember is that this is the juxtacrine or contact dependent signaling cascade. Here, this is the ligand, this is the receptor, and no doubt, just to show them, they are being shown separate. Otherwise, they are very much close to each other. So the kind of signaling where the two cells are most closer, that will be contact dependent or juxtacrine signaling, and paracrine will come thereafter. In the paracrine also, cells are nearby, but still they are bit far. In this, they are the two cells which are communicating with each other. They are very much close to each other. This fact is asked in so many exams. Now here it is an example of a particular question, that which of the following is correct. Now this particular question I have placed here to give you the summary of whatever we have taught so far. So in this particular question, because of the time constraint, I'm not looking into all the options and telling you what it is that you have to work out. But here, all these facts are correct, and the answer is P, Q, R, and S. So all this particular question, this particular kind of questions will be asked in the net, so as to see whether you know completely or not. because when more than one options are right, it will put more pressure on you. So such kind of questions are there in the CSIR net. Now, the properties of signaling pathways are that it will not just convey the message, it will amplify the initially small signal into a huge response. Signaling is done by few micrograms of signaling molecules, but the effect that will be generated might be few milligrams of you know metabolite, or so many cells dividing, so the effect can be huge. This is one of the important property of signal transduction pathway. Another one is it's not a single unidirectional thing that is happening. As so far we have looked into the thing, it will result into so many networks which are being activated. So it is not about express highway, it is about so many things which are being activated by a single ligand molecules as it will move down the signaling pathway. And the third and the most important feature or property of signal transduction pathway is it is remarkably specific and exquisitely sensitive. Even the picomolar concentration of the ligand or receptor will bring about the effect. And the particular ligand will bind to a particular receptor only. It's not like that कि message आया है कि भाई इंसुलिन जो है उसका प्रोड्यूस करना है, उसकी बजाय ग्लूकागन प्रोड्यूस हो जाए, ऐसा नहीं होगा. इंसुलिन का अगर काम है कि इट विल स्पीड अप the protein synthesis, lipid synthesis, gluconeogenesis, right? So it will do those things only. It's not like that it will start doing something else. So it is remarkably specific in its action. And that particular specificity again is being achieved by bimolecular association reaction and the molecular complementarity between the ligand and the receptor and the signaling molecule down the line. Most of them are non-covalent bonds, non-covalent forces, which individually are weak, but cumulatively they become very strong, as in the case of antigen antibody or epitope paratope interactions. Now, so far we have discussed that signal to receptor to transducer to effector, the response. This is, in a nutshell, is the summary of the signal transduction pathway we have discussed so far. From upstream to downstream, it starts with signal molecule, that is ligand, conveyed to receptor. Receptor will bind to it. It will pass on the message to transducer. Transducer will loop in the effector. Effector will be having secondary messenger. Secondary messenger will be having protein kinases, and protein kinases will bring about the effect by modulating the action of various enzymes and modulating the gene transcription, thereby bringing about the response. So this is, so far we have discussed. So another aspect of cell signaling is the amplification. As you can see in this particular slide, that one particular molecule of epinephrine resulting into a huge amount of product. This amplification is one of the important feature of cell signaling pathways. Now, why cell signaling is needed? It is needed for variety of purposes within the cell, because by means of its diversification, by means of its speed, by means of the kind of response that it can generate, the cell signaling plays a very important role in various aspects of the cell, be it a cell cycle or anything else. Now, this is example of two kinds of ligands. One is the signaling molecule, which is soluble. Another ligand molecule is the membrane bound receptor. So this is an example of contact dependent or juxtacrine signaling. The kind of questions that are being asked, I have shown you on the slide. Which of the following hormone passes through the cell membrane, binds to the intracellular receptor and then activates it? So obviously it has to be a steroid or estrogen hormone. So this kind of questions are being asked. Now we are coming to the types of receptors. And the types of receptors, maybe, two we have discussed, that one is present on the membrane, or this particular receptor is called as serpentine or GPCR receptors, because here the polypeptide is traversing to and fro uh, through the membrane at least 7 times. So that is why it is, since it is having a feature like snake, so it is called as serpentine receptor. There comes the receptors which are present in the cytoplasm or nucleus. So they are like steroid receptor or intracellular receptors. They are receptors which are like gated ion channels. They will allow the movement of certain ions through them by opening and closing. There might be receptors which are adhesion kinds. They get adhered to each other. Sometimes receptors are there which are, having an enzymatic activity on the cytosolic side. So such receptors are known as receptors with enzyme activity. So these are the various kinds of receptors depending on the different kinds of ligand molecules. So these are the type of questions which are being asked. The mechanism of signal transduction by steroid hormone differs from amine and peptide hormone because, so the fact that we have discussed is being asked here. So this is, it will not be a simple thing that they will ask. They will be very specifically ask you about the fact whether you know or not that only lipophilic can get into the cell. Now these are the 7 major kinds of membrane receptors, out of which the first one, this is GPCR, G protein coupled receptor. Another is receptor tyrosine kinase, and third one is cytokine receptors. These 3 receptors are very important, and they are being asked in various competitive exams more preferably than others. Now cytokine pathway is being governed by JAK-STAT pathway. So now they are asking, JAK-STAT pathway plays a central role in, so obviously cytokine mediated signaling cascade. So this is again a question taken from NET. Cytokine receptors, so again they are asking, have carboxy terminal domain which shows kinase activity, or are single membrane transmembrane polypeptide provide binding sites for non-receptor tyrosine kinase, or provides docking site for binding of RAS protein. So actually they are having this P and R for this one. So which of the following is are correct about JAK-STAT pathway? So actually all the given statements are correct when you will go through the cytokine pathway. Cell surface receptors may be any one of the following except transcription factors, because transcription factors are there in the nucleus. Now we are coming to G protein coupled receptor. Now this G protein coupled receptor is very important category. Variety of pathways, variety of signaling cascades in the cell are being governed by GPCR. And this is the domain which will bind to the ligand, and this is the domain which will convey the message into the cell. So the questions which are being asked, like G protein will bind to what, ATP or GTP? So G protein will bind to GTP and GTP both, but in the active form it will bind to GTP. So these GPCRs are very important and ubiquitous class of eukaryotic receptors, almost 700 types, various signaling cascades, our olfaction, visual, our hormonal, all those things are being governed by one or the another GPCR. And the intracellular domain is coupled to a trimeric G protein that is comprised of alpha, beta, and gamma subunit. So they are being called as G alpha, G beta, and G gamma. When the G alpha subunit of this trimeric G protein is bound to GTP, it is active or on. When it is bound to GDP, then it is off or inactive, as you can see in this particular diagram. This is a trimeric G protein, and hence this particular receptor is called as GPCR. So moment it is active, it will bind to it, and you can see this G alpha and G gamma, they are bound to the membrane. G beta is not. So sometimes that fact is also being asked in various examination. So now we are discussing how the signaling is done via GPCR. So moment the ligand bind to the receptor, it will become active. When it becomes active, there occurs a conformational change in the receptor. So this entire G protein will become accessible to it. Moment it become accessible to it, its G alpha unit will have increased affinity for GTP and will become active. When it becomes active, it will dissociate from G beta and G gamma and it will go and bind to effector, thereby activating it. Effector can be adenylyl cyclase or guanylyl cyclase. When it becomes active, it results into the synthesis of secondary messenger. And that secondary messenger will activate protein kinases, and that is how the effect will be brought about. And this entire G protein, G alpha, which is bound to GTP, it will be inactivated by autohydrolysis. And when it is bound to GDP, it will be inactive. There will be no affinity for this effector. It will come back to G beta and gamma, and this effector will become inactive then. So if ligand is not there, again this entire cascade will stop, and this trimeric will come into its native or original conformation. So this is how the entire cell signaling cascade is being governed in the case of GPCR.